May 18, 2020. Moderna Inc., a clinical-stage biotechnology company, announced positive interim clinical data of mRNA 1273. mRNA 1273 is a promising vaccine against SARS-CoV-2, the new coronavirus, encoding for a prefusion-stabilized form of the spike S protein. The spike S protein was selected by their scientists, in collaboration with investigators from Vaccine Research Center (VRC) at the National Institute of Allergy and Infectious Diseases (NIAID). The first clinical batch, which was funded by the Coalition for Epidemic Preparedness Innovations, was completed on February 7, 2020, and underwent analytical testing. It was shipped to NIH on February 24. 42 days, from sequence selection. The first participant in the Phase 1 study of mRNA 1273 was dosed on March 16. 63 days from sequence selection to Phase 1 study dosing. On May 6, the U.S. Food and Drug Administration (FDA) completed its review of the company's investigational new drug application for mRNA 1273, allowing it to proceed to a Phase 2 study, which is expected to begin shortly. On May 12, the FDA granted mRNA 1273 Fast Track designation. Moderna is finalizing the protocol for a Phase 3 study, expected to begin in July 2020. Here is the timeline of their response through May 7, 2020. On January 11, 2020, the Chinese authorities shared the genetic sequence of the novel coronavirus. On January 13, 2020, the U.S. National Institutes of Health and Moderna's Infectious Disease Research Team finalized the sequence for mRNA 1273, the company's vaccine against the novel coronavirus. At that time, the National Institute of Allergy and Infectious Diseases disclosed their intent to run a Phase 1 study using the mRNA 1273 vaccine in response to the coronavirus threat, and Moderna mobilized toward clinical manufacture. The manufacture of this batch was funded by the Coalition for Epidemic Preparedness Innovations. On February 7, 2020, the first clinical batch, including fill and finishing of vials, was completed. A total of 25 days from sequence selection to vaccine manufacture. The batch then proceeded to analytical testing for release. On February 24, 2020, the clinical batch was shipped from Moderna to the NIH for use in their Phase 1 clinical study. On March 4, 2020, the U.S. Food and Drug Administration completed its review of the investigational new drug application, filed by the NIH, for mRNA 1273, and allowed it to proceed to begin clinical trials. On March 16, 2020, the NIH announced that the first participant in its Phase 1 study for mRNA 1273 was dosed. A total of 63 days from sequence selection to first human dosing. This Phase 1 study will provide essential data on the safety and immunogenicity of mRNA 1273. Immunogenicity means the ability of the vaccine to induce an immune response in participants. The open-label trial is expected to enroll 45 healthy adult volunteers ages 18 to 55 years, over approximately 6 weeks. On March 23, 2020, Moderna filed a current report on Form 8K, which included, among other things, information regarding the potential timing of the availability of a vaccine against COVID-19. The company reported that the Phase 1 study is proceeding following the protocol under the direction of NIAID. The company further reported that while a commercially available vaccine is not likely to be available for at least 12 to 18 months, it is possible that under emergency use, a vaccine could be available to some people, possibly including healthcare professionals, in the fall of 2020. Any emergency use would be subject to authorization by the appropriate regulatory agencies, based on the emergence of clinical data for mRNA 1273, that would support the use of the vaccine before licensure. Also, the company confirmed that it is scaling up manufacturing capacity toward the production of millions of doses per month, in the potential form of individual or multi-dose vials. As has previously been disclosed, the ability of the company to make millions of doses per month is contingent on investments in the scale-up and further build-out of the company's existing manufacturing infrastructure. On March 27, 2020, the NIH announced that Emory University in Atlanta would enroll healthy adult volunteers ages 18 to 55 years, in the Phase 1 study of mRNA 1273. On April 16, 2020, Moderna announced an award from U.S. government agency, BARDA, for up to $483 million to accelerate the development of mRNA 1273. This award will fund the development of mRNA 1273 to FDA licensure and manufacturing process scale-up to enable large-scale production in 2020 for pandemic response. Moderna plans to hire 150 new team members to support these efforts. Moderna also announced that the lead Phase 1 study of mRNA 1273 has completed enrollment of 3 dose cohorts: 25 micrograms, 100 micrograms, and 250 micrograms, and is expanding to an additional 6 cohorts of older adults and elderly adults. On April 27, Moderna announced that it submitted an IND to the US FDA for Phase 2 study of mRNA 1273. On May 1, Moderna and Lonza announced a worldwide strategic collaboration to manufacture mRNA 1273 to enable manufacturing of up to 1 billion doses per year. On May 6, Moderna filed a current report on Form 8K, which included an interview published by National Geographic with Anthony S. Fauci, director of NIAID. The report described his assessment of the results of certain preclinical testing related to the ongoing Phase 1 clinical study of mRNA 1273. On May 7, Moderna announced that the FDA completed its review of the company's IND application for mRNA 1273, allowing it to proceed to Phase 2 study, expected to begin shortly. Moderna is finalizing the protocol for Phase 3 study of mRNA 1273, expected to begin in the early summer of 2020. The following short video from Moderna's website summarizes how the potential vaccine against COVID-19 was developed and how it works to help humans create self-immunization. Keep watching and enjoy.
It all started with isolation of the virus from an infected patient in China. The coronavirus was sequenced, and its genetic information was uploaded to a public database. Working together, scientists at the NIH and Moderna identified the sequence for a key protein on the surface of the virus, called the spike protein, as a good vaccine candidate. The instructions for making the spike protein were then encoded into an instruction molecule called mRNA, which could be administered directly to patients as a vaccine. The vaccine is injected just like other vaccines. The mRNA is taken to immune cells, where it instructs cells to make copies of the spike protein, as if the cells had been infected by the coronavirus. Other immune cells, shown here as red and orange figures, are then able to learn about the spike protein and develop ways to protect the person if they ever come into contact with the actual coronavirus. What makes this approach different is that you don't need to make the virus itself to make a vaccine, a time-consuming and intensive process. Instead, you use the information from the virus and administer the information directly to the patient. Essentially, the patient makes their own vaccine. This cuts out the middleman. Using this approach, NIH and Moderna were able to go from sequence to a vaccine ready for human testing in record time, just 42 days.